Hello everyone and welcome to this webinar co-produced by the CRISPR Journal and Genetic Engineering and Biotechnology News. Our presentation today is entitled A CRISPR Vision for the Future of Cell Therapy. It's sponsored by TriLink Biotechnologies. This is the second in a three-part webinar series sponsored by TriLink. I'm your host, Kevin Davis, the executive editor of the CRISPR Journal, which has been publishing research in genome editing and CRISPR biology for some 2.5 years. I'm also editor-at-large for GEN, Genetic Engineering and Biotechnology News, which has been covering the biotech industry for about 4 decades. Let's meet our special guest speaker for today's webinar. I am delighted to welcome TJ Cradick, a leading gene editing researcher with almost 2 decades experience in the therapeutic use of engineered nucleases. Since 2012, TJ has focused on CRISPR-Cas9 gene editing technology development, leading efforts to improve therapeutic activity and specificity, and developing the bioinformatics tools that enable these studies. TJ is currently a consultant and advisor in gene editing, gene and cell therapy, and bioinformatics, having recently left CRISPR Therapeutics, where he was head of genome editing and the second US-based employee. His group at CRISPR Therapeutics initiated the hemoglobinopathies program, now in clinical trials. He previously served as director of the protein engineering facility at the Georgia Institute of Technology and Emory University. TJ studied at MIT and UCSF and holds a PhD in molecular and cellular biology from the University of Iowa. Before we get underway, we hope you'll submit your questions for the Q&A session at the end of today's presentation. We'll try to get to as many of your questions as we can in the time allotted. Please hit the Ask a Question button that you will see at the top of your screen and hit Submit. All right, let's get today's presentation underway. TJ, thanks so much for joining us. We're really looking forward to your remarks. Over to you.
Thank you, Kevin, for the introduction. Hello, I am TJ Cradick. I'd like to thank Genetic Engineering and Biotech News, the CRISPR Journal, and TriLink for inviting me today to present on a CRISPR vision for the future of cell therapy. I must disclose that I'm a patent holder, consultant, and advisor in CRISPR, gene editing, gene and cell therapy, and bioinformatics. I'll start today with an introduction, including some definitions. Cell therapy is defined as transferring live whole cells with the relevant function into a patient for the treatment of a disease. The cells can be injected, grafted, or implanted. Some cell therapies use the patient's own cells that are genetically modified. Other cell therapies use healthy donor cells that can also be genetically modified. Gene therapies are broadly defined as the introduction, removal, or change in the content of a person's genetic code to treat a disease. These gene therapy strategies correct or modify gene expression or produce new or modified proteins. Each of these gene therapy strategies involves the administration of a specific DNA or RNA into the appropriate cells, usually in a carrier or a vector to allow uptake. A subset of gene therapy is gene replacement therapy. In gene replacement therapy, you give the cells a new working copy of the missing, mutant, or non-working gene. Another subcategory of gene therapy is gene editing, where there are changes to the existing gene and they can be used to correct mutations where they occur. These changes can include insertions, deletions, inversions, or other edits that might be used to treat the disease. Gene editing can occur inside or outside the body. Both gene and cell therapy have the potential to treat the underlying cause of both genetic and acquired diseases. Gene therapy and cell therapy therefore work hand in hand and are often combined to treat various genetic diseases. Gene therapies can be divided into two groups based on where the cells are modified, either in vivo or ex vivo. In the coming slides, I borrowed some cartoons from the ASGCT website, which I'd recommend for their introduction. In vivo gene therapies include genes introduced into the patient's cells within the body. Ex vivo gene therapy includes removing the patient's cells, treating, adding, or editing the genes, and then reintroduction. For this reason, many cell therapies include gene therapy, such as the introduction of new expression cassettes. Many cell therapies also take advantage of the recent advances in gene editing to correct mutations, knock out genes, or make other changes to improve the function of the resulting cell therapies. In the remaining slides, I'll outline some current and future cell therapies. As we continue to improve our editing and cell culture techniques, we're seeing an expanding repertoire of cell types for cellular therapies. Cell therapy treatments can include isolation and transfer of specific stem cell populations, the administration of effector cells, induction of mature cells to become pluripotent cells, and the reprogramming of mature cells. I'll next provide some illustrative examples of cellular therapies.
I'll start with an early and still common cell therapy, bone marrow transplants. In 1956, Don Thomas treated a leukemia patient with their twin siblings' bone marrow. Bone marrow transplants are the most common and well-established cell transplantation therapy, with thousands requiring life-saving bone marrow transplants annually.
Successful bone marrow transplants require the injected cells to home into the afflicted bone marrow, integrate, proliferate, and recover to reestablish its biological function. Many of the other therapies that we discuss and are in current use require similar steps for the cells to home, integrate, proliferate, and recover. Chimeric antigen receptor T cells, or CAR T cells, present a great example of how gene therapy, including gene editing, can combine with cell therapy, and they present a great example of how CRISPR and related technologies are changing the future for cell therapies. The excitement about CAR T therapies was even heightened as two new ex vivo gene and cell therapy CAR T therapies were recently approved. The first ex vivo therapy, Novartis' Kymera, was approved by the US FDA in August 2017 to treat acute lymphoblastic leukemias. A few months later, in October 2017, Yescarta by Kite Pharma was approved for some types of large B-cell lymphomas. Let me start by describing chimeric antigen receptor, or CAR, modified T cells. The normal T cell is depicted on the left. On its surface, there is a T cell receptor, and that's binding to a peptide on the surface of the tumor being displayed by the major histocompatibility complex 1 protein. In the chimeric antigen receptor genetically engineered T cell to the right, there is this CAR complex on the surface that is specific for the antigen depicted, CD19, that is on the tumor cell. This table provides a comparison of some of the many different CAR T therapies. The first two columns are Yescarta and Kymera, as mentioned, plus a range of others. I'm bringing this chart to just exemplify that there are a number of facets within each of these therapies, including a range of different CAR constructs. So by changing the gene cassette that's introduced, one changes the specificity of this single chain binding epitope, such that one changes the antigen to which these CARs are directed. They also differ between the co-stimulatory domains that are included with this construct. They differ in the viral vector or the gene editing means, and on the bottom, whether they're autologous or allogenic donors, which I'll describe a bit more. The next cartoon shows a range of different co-stimulatory pathways that could be employed by CAR-Ts to bring up the point that there could be multiple gene editing opportunities to make these different generations or different types of CAR-T cells. This next cartoon follows on this theme with examples of gene editing strategies to optimize the CAR-T functions. In the top left are a number of different edits that can be employed to increase persistence. In the top right, a number of cellular targets that can be edited to change the tumor microenvironment. Below are a number of different strategies to change the potency of the CAR-T cells. And in the bottom left, a number of changes that can be made to change the homing. And you see from the numbers on here and the different opportunities is that it presents a number of ways that one can multiplex edit. One can do multiple different edits at the same time to enact changes for each of these. In the next few slides, I'll describe the range of possible sources or donors for cell therapies. In autologous cell therapy, the patients receive their own cells after engineering. In allogeneic cell therapy, the source of the cells is a healthy donor. These previously manufactured cells are therefore referred to as off the shelf. In xenogeneic cell therapies, the cells are from another species. One example is engineered pig cells or organs that are available for transplantation. This slide presents an overview of the steps in autologous cell therapy as described before, where the cells are harvested from the patient and then gene edited with GMP, Cas, guide RNAs, viruses, etc., before being transplanted back into the patient. Obviously, there's quite a number of steps in this process, including QA, QC, monitoring of the reagents as they're going in and the cells after they're produced. But wanted to depict it this way, as we'll be comparing it to allogeneic shortly. I've included this slide to indicate that these autologous CAR-Ts have many more steps, including the quality control in the top right, and also to drive home the point that each CAR-T product is custom-made and patient-specific. I've used the same simplified cartoon to illustrate the allogeneic cell therapy. The top left, though, is a healthy donor whose cells are apheresed and provided for editing. This allows a scale-up in manufacturing before diagnosis, so that once the patient presents, there's not the time delay. I'll describe this a bit more in the following slide. The next cartoon lists a number of the advantages of allogeneic versus autologous CAR-T therapies. On the top is the autologous process. I've added to this diagram a line marking the diagnosis. For the autologous process, everything starts after diagnosis. So that's when the cancer treatment decisions go on, the patient is apheresed, the manufacturing begins, and then the individual CAR-T therapy happens after manufacturing. And that's contrasted with the panel below that depicts the allogeneic process. So well in advance, healthy donors are apheresed. We have a scalable manufacture of 100 plus doses or batches that can treat a number of patients. There's quality control to ensure the manufacturing process, and these are stored so that at diagnosis, a patient receiving the allogeneic therapy does not have to wait for the manufacturing. So it saves time with these 6 patients. It also enables a number of quality assurance steps and the possibility for cost savings and market access. Here I've chosen a quote from a recent review paper that states that 2 recent scientific findings paved the way for a bright future of cell therapy. The first they listed is the development of induced pluripotency by Shinya Yamanaka. Using a combination of transcription factors, Yamanaka et al. Were able to transform stem cells in adult cells into induced pluripotent stem cells, iPSCs, that behaved like ESCs. The other was the application of CRISPR-Cas for genome editing and engineering. This cartoon depicts the clinical strategy for human-induced pluripotent stem cell-based gene and cell therapy. In figure A to the left, the patient is depicted in gray, and going clockwise, cells are isolated from the patient. A number of different treatments, including either zinc fingers, talons, and CRISPR, are depicted at the top right to make gene-corrected human-induced pluripotent stem cells. Figure B shows a number of factors that go into play, including some quality control screens and haplotype and banking. This is followed by gene-corrected human-induced pluripotent stem cell-derived target cells that can be transplanted back into the patient.
Another cell therapy opportunity is to use induced pluripotent stem cells that have been engineered to be resistant to HIV, similar to the way the Berlin patient was resistant to HIV tropic for the CCR5 receptor. In this paper, human stem and progenitor cells had their CCR5 gene edited. The resulting T cells were resistant to HIV challenge in the mouse model. This slide describes ongoing efforts among many groups to make universal induced pluripotent stem cells. This paper describes using multiple edits to inactivate MHC class 1 and MHC class 2 and knocking in a transgene to overexpress CD47. These hypoimmunogenic iPSCs retain their pluripotent stem cell potential and differentiation capacity. The title of one news article on this stated, "CRISPR gene editing makes stem cells invisible to the immune system." And obviously this is important if we want to continue to use universal donors for off-the-shelf therapies. This figure represents current and possible therapies for diabetes. Panel A on the left is standard therapy, exogenous insulin administration. On the left, the use of multiple shots per day, and on the right, an insulin pump, which can also have a continuous glucose monitor to work as a bit of an artificial pancreas. Although we have engineered human insulin available, insulin from pigs and cows was prevalent and is still marketed. Panel B represents replacement therapy, whole pancreas transplantation, or islet cell transplantation. The islet cells are the part of the pancreas that's responsible for making insulin in response to rising blood sugars. Panel C represents stem cell therapy. The discovery of the great differentiation potential of stem cells has allowed progress on obtaining sufficient quantities of the pancreatic beta cells, and they can be derived from mesenchymal stem cells, MSCs as depicted here, or human pluripotent stem cells, including human embryonic and induced pluripotent stem cells, derived either from the patient or from donors. These hypoimmunogenic iPSCs have great therapeutic potential. A recent review quoted, "Finally, overcoming the immune barrier to transplantation would also provide an exciting new modality to treat autoimmune diseases such as type 1 diabetes," as on the last slide, and multiple sclerosis, where one particular cell type is attacked by the patient's immune system and needs replacement. But I also want to emphasize that making these hypoimmunogenic iPSCs also allows the opportunity for future edits, to have multiple edits to improve the cell function. This brings us to the next section of the talk, gene editing for cell therapy. I hope I've described why gene editing is a critical component of cell therapy, but next I'll go to a quote. "We realized early on that refined gene editing techniques were what was needed to take an allogeneic approach to CAR T-cell therapy."
So before getting into gene editing, let me start by describing the classes of engineered nucleases. There are a number of different types of nucleases. that have been designed to edit the genome. This is some simplistic cartoons, but gives you a feel for, starting on the left, meganucleases, B, zinc finger nucleases, and tau effector nucleases. You can see in cartoons B and C that each of these has two proteins that are designed to bind with correct spacing and orientation for the FOC nuclease to dimerize in the middle and cleave the intervening sequences. Each of these A, B, and C requires changing or designing new proteins for each target. Below is the CRISPR-Cas system, which I'll describe a little bit more. And certainly gene editing interests in publications and other readouts expanded dramatically after recent papers on CRISPR-Cas, one of which from 2012 was a programmable dual RNA-guided DNA endonuclease, an adaptive bacterial immunity. And this paper described taking this system and redirecting it towards your favorite gene or your therapeutic target. It also described the process of linking this tracer and CRISPR RNAs, which I'll go into in a moment. The key factor is that the protein remains unchanged. There are different Cas9 proteins, but you can continue to use the same one for each target. And again, this contrasts to zinc finger nucleases or tau effector nucleases, where one would design and reclone a new protein for each target. So the part that would change to go to a different target would be this guide RNA. And as described, there's two RNAs that could be linked together, and I'll jump to that next. In our constructs, we tend to use a single guide RNA that's made up to the two naturally occurring RNAs in the CRISPR-Cas system. The CRISPR RNA, which has been shortened and shown on the left here, is combined with a shortened version of the tracer RNA and connected by a short loop. So this CRISPR plus tracer are formed together to make the single guide RNA. And the tracer part remains unchanged for each target. Only the target site portion of the CRISPR RNA is changed. So you can see that the design of this is fairly straightforward. Once one identifies a target site, the part that's boxed there as the spacer is changed to redirect the protein to target that site. For that reason, one can screen a large number of sites fairly readily by changing this RNA. The cellular repair is part of the cell's natural DNA repair pathway. And so the part that we're putting in just causes the nuclease cleavage. And so the CRISPR cleaved DNA is repaired using these cellular pathways. And there's a range of different ways that cells repair their broken DNA, obviously, as that's so critical. But the two main paths, or the two big divisions that we describe, are the non-homologous end joining pathways, the down and dirty ways of getting those ends together, versus the homologous recombination pathways. As the non-homologous end joining pathway is preferred in certain cells and in certain parts of the cell cycle, I've made that font much larger here to remind us of that.
We can use these different repair strategies as we go after different disease indications, as I'll describe in the subsequent slides.
I'll start with this representation of the non-homologous end joining pathway, which summarizes a number of related DNA repair pathways for the DNA ends to be joined. Cells have a number of means for repairing these loose ends in hopes of keeping their chromosomes together and DNA intact. When the DNA is cut, it is often repaired perfectly, which reforms the initial sequence. It is hard to know how often this occurs, as the process is hard to observe, as the sequence is same as the uncut. The sequence is then available to be bound again, and the cut-free ends are available for repair. The various repair assays, including the cutting assays and next-generation sequencing, observe this cutting as the accumulation of these misrepairs. non-homologous end joining and related pathways are often successful for knocking out gene expression, as many of the misrepairs lead to the loss or gain of bases, as I'll show at the next slide. Knocking out particular sequences, including transcription factor binding sites, can also lead to changes and increase in expression. Next, I'll show a plot of the various sizes of insertions and deletions we observed with a series of guides targeting around the sickle cell mutation in beta globin. The sizes detected is based on the assay. Here we use topo cloning and Sanger sequencing of the individual PCR products. Insertions are less common and shown to the right of the 0. The frequency is in percentage of the indels is shown to the right, and the size of the individual insertions on the right or deletions to the left is below each bar. I put a green box around the common single base deletion that we tend to see, and we can also see in this one a high frequency of single base insertions and a range detected using this assay. In addition to having single DNA breaks, one can put in multiple guides, and in this case, these 2 specific DNA breaks can result in non-homologous end joining or related repair, making a defined deletion or an inversion. In addition, there's the homologous recombination or homology-directed repair DNA repair pathway. Here again, we have a specific DNA break through our engineered nuclease. But for homologous recombination, we supply a donor DNA. And this can be single-strand oligonucleotides or longer. It can be double-stranded DNA or plasmids we used to use. Often we'll see data presented from adeno-associated virus, which is a very effective donor DNA. But each of these different kinds of donor DNAs shares the feature of homology arms, or sequences that match upstream and downstream of the target site. Here I've depicted the change as this green rectangle. Once it's repaired by the homologous recombination pathways, we can correct point mutations, such as we might want to do for sickle or cystic fibrosis. We can put in multiple exons if we want to change and correct disease caused by a series of different mutations. We can knock something into the safe harbor location, as is often done for some of the liver indications, or we can put in a different transgene into another site. Another means of repair is oligo insertion. And here, as opposed to homology-directed repair, we think of the oligo going into the cut site, which can be used for correcting point mutations, knocking out, or including a binding site, such as a transcription factor binding site. An additional means of DNA repair is using microhomology-mediated sequences and microhomology-mediated DNA repair. I've indicated 2 short homologous sequences flanking a target site here as these purple arrows. Again, this can lead to a defined deletion or knockouts when they're repaired by the microhomology-mediated end joining paths. I'll again return to the same indel spectra, this time focusing on the area with the purple rectangle around 9 nucleotide deletion. And this we attribute to the microhomology found with 9 nucleotide spacing. When this is repaired, we see this high frequency of these bases being removed, as an example of microhomology-mediated end joining and loss of these sequences. Alternate ways of gene editing. And one that has gathered much excitement is base editing, where instead of having a nuclease cut both strands, there's a nickase that cuts one of the strands, and the base editing is supplied with deaminase and possibly other factors, such that you can drive specific DNA changes, such as C to T or A to G. And one can look at the numbers of different single base nucleotide changes that can be corrected by this, both to correct disease-causing mutations and also, as described before, for knocking out particular genes, putting in stop codons, changing splice sites, etc. Another recently published method is prime editing, which uses a fusion protein consisting of a catalytically impaired Cas endonuclease fused to an engineered reverse transcriptase enzyme, and uses a prime editing guide. And this longer guide I've boxed in the top of the figure. And this works like a homology directed repair donor DNA, and that it allows for specific editing and inclusion of these sequences. And I'll show that in the bottom right with this box again. And so this has been developed the last year, and we're continuing to see that this method has been used by others and published further. One of the aspects that separates CRISPR from the other gene editing systems is our ability to multiplex. As a new protein set isn't needed for each target as they were for talons and zinc fingers, the same protein can be used and redirected by individual guide RNAs that can be supplied for each new additional target site. A number of papers are out describing multiplex editing using CRISPR-Cas systems and CPF1 in a range of different organisms. I'll return to this cartoon to make the point that sometimes the term multiplex indicates only two guides, but in general it's two or more specific DNA breaks. In this example, they all are repaired by non-homologous end joining. Multiplex editing can include a range of different DNA repair pathways. Here, going back to the cartoon, I show the donor being added. And so in this cartoon example, we have two knockouts because of repair by NHJ, and the inclusion of this donor DNA allows functional gene correction, or in the case of CAR T, insertion of a chimeric antigen receptor expression cassette. I've included this cartoon to show one path for CAR T multiplex gene editing. On the left, a virus will deliver the CAR construct into the genome, and on the right, I've shown nucleases to allow precise knockouts or addition of other genes. CRISPR-Cas9 systems have recently emerged as a simple and efficient way for multiplex genome engineering of CAR T cells by combining lentiviral delivery of the CAR expression cassette and electrotransfer of Cas mRNAs and guides targeting endogenous T cell receptor, beta-2 microglobulin, and PD-1 genes simultaneously to generate gene-disrupted allogeneic CAR T cells deficient in expression of TCR, HLA class I, and PD-1. In another recently published example of multiplex CAR T editing, we're using Cas9, but this case is a ribonuclear protein with guides specific for TRAC A, TRAC B, and PD-1, and again using lentiviral delivery of a T cell receptor. And here's a cartoon on the right showing these genetic changes and the resulting surface expression. I'll return to this cartoon, which I've modified to show on the left a donor DNA and nucleases being used to precisely insert the CAR construct. This paper did precisely that, using CRISPR-Cas guides to target the T cell receptor alpha and to allow insertion of their CAR expression cassette. In this mouse model, this provided uniform expression. This figure is from a paper that was using multiplex precise base editing in monkeys to model tyrosinemia by inserting mutations into the FA gene. They designed a single guide to introduce the W75X stop mutation in exon 4, shown in the top left. The PAM is on the far left of this figure, and the two changes where they're converting C's to T's would introduce this mutation. They followed on by looking at the possibility of editing two other sites. And part of the reason I chose this figure is that there are multiple nucleotides that could be changed in each of these windows that they're targeting. So in addition to having guides target 3 different locations, there's a chance for changing multiple nucleotides in each of these activity windows. So that's certainly an aspect of base editing that continues to be researched in order to narrow that window, or as in this opportunity, to have multiple base changes at each site. To continue with our examples of T-cell engineering, this time using multiplex base editing, the ribonuclear protein is provided along with guides specific for TRAC, B2M, and PD-1, as described before, along with lentivirus delivery of the TCR. So here we're seeing similar knockouts of these genes and delivery of the construct, but this time using multiplex base editing. After showing examples of multiplex nuclease editing and multiplex base editing, I have this cartoon in to describe what I've dubbed multiplex multi-mode gene editing, to alert of the possibility of using both systems simultaneously or in succession. One can knock out a number of different genes either way and provide donor DNAs allowing functional gene correction or insertion of expression cassettes, as with the CAR-Ts. I think that going forward, we'll see many examples of further combinations of different methods. It has been very exciting to observe the therapeutic effect of these gene-edited cell therapies. Next, I'll describe some gene editing assays that have been developed to observe the genetic changes in the cells.
CRISPR-Cas, as well as each of the nuclease systems, has the possibility of off-target events at sites similar to the target site. In 2013, our group at Georgia Tech, our collaborators, and others noted that some guide RNAs can cleave unintended sites of the genome if they were sufficiently similar to the guide RNA. This work has led to a better understanding of the tolerance for mismatches and bulges, which has enabled better bioinformatic modeling and shown the importance of careful choice of the target site. It also has emphasized the need to test after nuclease use to ensure specificity.
To review the possibility of off-target cleavage, I'll go back to a paper we published with Gang Bao's group characterizing guides targeting near the sickle cell mutation in beta globin. The cleavage of beta globin, or HBB, is shown on the left. On the right, we also see a similar site in delta globin, or HBD. Guide RO3 on the top only has one difference between the site in beta globin and the site in delta globin. That one mismatch is shown in red, 11 bases from the protospacer adjacent motif on the right. This small data set shown in this figure exemplifies the trend that possible off-target sites with mismatches closer or more mismatches tend to have less off-target cleavage. Compiling this type of data has allowed us to use bioinformatic programs to help pick target sites that may have less chance for off-target events and help identify sites for further testing when we treat the cells with these nucleases. So to ensure specificity, we use these bioinformatic tools to help pick favorable target sites, but we always have to assay and test them and test the treated cells for possible off-target events. We also want to assay for unintended on-target editing events and for possible chromosomal rearrangements. These are in addition to the type of normal genotoxicity and cellular toxicity assays that might reflect adverse events from the treatments or the editing steps. So these possible off-target effects, and each of these assays tells you putative sites or sites that might have off-target events going on, and therefore that's why we always sequence to determine or check if there's editing and validate the amounts. And so these off-target events result from the cutting or editing at sites other than the intended target. The effects can include insertions, deletions, or substitutions, and can also include chromosomal rearrangements or large deletions. So there's a number of ways to quantitate or measure these editing effects. And these can include next-generation sequencing, droplet digital PCR for absolute quantification, a range of long PCR methods, or other assays. And there were a number of sessions at the ASGCT recently, including one I was on where Luca Pinello described his lab's development CRISPRCas for processing next-generation sequencing, and A.L. Hendel described a range of different methods for quantitating editing effects. So these are some of the factors that need to be weighed as we're looking to optimize our curative cell therapies. And we want to look at the intended edit percentage, both the number of alleles changed and the number of cells. Again, we want to look at the possible unintended edits and effects. There's certainly a greater effect by changing a nucleotide in a coding sequence than an annotated sequence that a single nucleotide change might not be important. And again, we want to look at genotoxicity, the cellular properties, and a critical factor is how well these cells engraft into the model organism or the patient, and therefore it seems that we need to keep an eye on this in our processes so that we maintain high levels of editing and high levels of engraftment. I'd like to thank those with whom I'm currently working, former collaborators, particularly those from Georgia Tech, whose data I presented today, and those who published the papers that I outlined in this talk. I hope you can check those to find out more information on their work. At this point, I'm available for questions and discussion.
Thank you very much, TJ. That was just a great presentation. We're still looking for your questions, so please enter them at the Ask a Question button at the top of your screen, and we'll get to those in just a couple of minutes. We'll be archiving today's webinar, a quick reminder, so you can view it on demand or share the link with your colleagues who were unable to join us for the live presentation. Okay, I think it's time for the Q&A session. So let's get back together again with TJ and just give us a couple of moments to transition into the Q&A session.
Thank you very much, TJ, again, for a masterful presentation and a TriLink for their sponsorship. Questions have been flooding in. So, TJ, we've got about 15 minutes. Let's see how many we can get through. And for those people who've asked questions that we don't have time to cover before we get to the top of the hour, TJ's given his email in his closing slide, and I'm sure he'd be happy to take questions offline if we're unable to address them all. So, TJ, if you're there, we're going to start. I often hear it's the first question about changing the gene, but rarely about editing the promoter. Promoters mutate at a similar level to genes, presumably. Do you know of any groups or approaches that are paying attention to editing the structure of promoters or other non-coding regions?
That's certainly an exciting element. We've, as many people are aware, have been targeting disease-causing mutations in the coding region. There's been demonstration of the importance of targeting transcription factor binding sites. And so the question is, if one was hoping to bash or knock out a particular promoter, whether that is as effective and would have as high a success rate. Some regions of the promoter, a single base change might not be as dramatic as changing the code, putting in a stop codon. But certainly these kind of strategies have been proposed, and it's just a question of how well you can have a NHEJ type change, as we discussed in the talk, and knock out a promoter. So that's part of the reason that people have been leaning towards either going to transcription factor binding sites that have dramatic changes, or often if they want to knock something out, to knock out the coding region. Same goes with trying to correct a promoter that might not be working.
Very good. All right, let's rattle through some of these to make up for time. What's the failure percentage of CRISPR? I'm not quite sure if the question is about it generally or, but take that question however you wish.
I'll try and address it on a couple levels. One is that CRISPR is a very robust system. If you look at 100 guides that are tiling a region, a very high percentage of them will cut and cut with high efficacy. Then the question is, how well can you, let's say, knock out a gene? And that it depends on doing some analysis to pick your target site properly, but we see a very high success rate. And the third part of the question might be, how well is this working in clinical trials? And I think that's what's exciting right now, that we're underway with clinical trials, and we'll get to see what the cure rate is as we move into patients hoping to cure their diseases.
Yeah, we'll come back to clinical trials a bit later. Is CRISPR now readily available around the world, or is it still in the research phase?
No, I think that's a great question. I remember in the days of old with zinc fingers, there were a number of papers, maybe some people in the audience published on the difficulties in assembling and designing zinc fingers. When we got to tau effector nucleases, those were easier to design, but more difficult to clone because of the large size of these proteins that have to be put together, as I showed in one of the early slides. And so there was kind of
a renewed enthusiasm, although it was difficult to jump into for many groups. Whereas CRISPR, particularly starting in 2013, one could go to Adgen or right to Feng Zhang's lab and get one of the early plasmids, PX330. And it would take you a very short amount of time to look for a PAM site, pull out the target site next to it, order a pair of oligos, kind of anneal and throw them into the plasmid, and then you could go in. And so if one particularly uses these resources and academic collaborators, one can get these reagents very readily throughout the globe.
And so Adgen is the first port of call for any researcher who's looking to start off in this area?
Certainly, they are the go-to for all academic and probably nonprofit groups, and that's what they've been just amazing at dealing. So yes, I think that people should certainly have them on their radar. And I also want to give a shout out. They've got a great introductory page that I remember a few years back sending a lot of people to read up some more on certain aspects of CRISPR-Cas and gene editing.
Yeah. Well, you gave a masterful kind of tutorial on cell therapy and genome editing, but one topic you didn't get round to is gene drives. But this question is a good one. What do you perceive the future use of CRISPR-Cas9 applications in the gene drive context, for example, to remove a lethal genetic defect from the global population? I think most common applications that we hear about these days, it's potentially with regard to eradicating malaria. The idea scares me, says the questioner, but what's your view?
Well, I think that many of the questions that come up, as you mentioned, in this topic of going after particular mosquitoes that harbor malaria. And many times, just like the question about going to germline, are we cognizant of all of the risks that are inherent? And with many things, we want to balance the risk benefit. And so for here, the question is, if there's a lot that we don't know, how well can we make this risk benefit analysis? And I think that's one of the driving parts, besides that we lack some of the technology to move forward on it in this particular application, which is certainly a more complex endeavor than targeting the mosquitoes. And so I think that there's just a lot of questions and technical hurdles that remain to move forward on that.
Okay. Why, back to cell therapy, why are pluripotent stem cells used in therapy and not totipotent stem cells?
Certainly. I think this goes back to my question about if I know these terms correctly, totipotent is even further. It's encompasses also embryonic cell lines. And so I think that if we're not interested in moving towards germline editing, then it raises ethical and probably technical questions. I think have difficulty and have some technical challenges to overcome to get into the stem cells in general. And so trying to go into stem cells that provide other capabilities that we're not interested in might be as much as an ethical and a technical challenge.
Thank you. The next question, I'm going to riff on this question and ask you one of my own. But the question was, is there a therapy based on CRISPR approved by the FDA? And you've kind of touched on that with the hemoglobinopathies, and of course, Editas's Leber congenital amaurosis therapy. But I want to use that as a cue to ask you, you've had a fantastic front row seat for this first wave of commercial genome editing, CRISPR genome editing success and potential from your 4 or 5 years as head of genome editing at CRISPR Therapeutics. So I'm curious, with the first clinical trials underway, are we about where you thought we would be when you joined CRISPR Therapeutics 4 or 5 years ago? Are we ahead of the game? Are you disappointed with how we've only got a couple of trials FDA approved? What are the next big milestones that we should be looking for, and what trials are you and other people in the field watching or anticipating with most interest?
I think the first part of the question I can jump on more quickly, and that is, would you expect to be at this point? And I think anyone who's worked in molecular biology or small molecules or other therapeutic settings realizes it's just tremendous to be around on something you've worked on, getting into patients, and still being at the company or still being involved on the project. It's just an amazing part that in a handful of years, one can go from thinking about and choosing a target to developing the pipeline, to doing the preclinical and regulatory package, and then seeing it in patients. And I think that it's a testament to both the seminal work that was done in the past by many luminaries, to just the robust nature of this platform. So I think it is, you can't be surprised, I think, if you've been around long enough to see many things that take much, much longer and spend a long time in optimization. To jump to the second part of the question, you mentioned the CRISPR Therapeutics and EditOne trials, and I think those are very exciting. Certainly, the hemoglobinopathies is a tremendous target because of its significant worldwide burden, morbidity, mortality, and the genetic cure holds great promise. There's certainly other ways to treat some of the symptoms, but there's a heavy burden on patient care. So a CRISPR cure is very exciting. And so I imagine that, as I mentioned, we've seen some of the initial data, but I think as the European Hematology meeting is coming in just a handful of days, people can look to hear what CRISPR Therapeutics and Vertex, who are together on the study, will be announcing the 12-month follow-up data for the first patients treated in CLIMB 111, which is for transfusion-dependent beta thalassemia, and 6-month data for the follow-up for the sickle cell patient. And what the reason I want to kind of amplify that is it's exciting to be treating patients, and this goes to the first part of the question, because now we can see them actually receiving the medicine that we've been studying for quite a while, and see how they progress. And this is also exciting for the other trials that are forthcoming, because we've been modeling these diseases for years. And when I say for years, I mean going back to zinc finger nucleases and before. And so we'll learn a lot from these studies. And so I'm very excited about the CRISPR Vertex hemoglobinopathies. But as you mentioned, it's also exciting that EditOne has announced its first dosing for EditOne, which is for the treatment of Leber, as you mentioned. And I think the reason that that's also very interesting is that it's promising treatment for a disease-causing mutation, but it's also in a different organ, and it's in vivo. So we're treating inside the patients. So I think there'll also be a lot that we'll learn, plus the excitement of getting into these patients.
Yeah. And TJ, you touched on base editing in your presentation. Obviously, base editing is still probably a little ways away from the clinic, but some very exciting and promising results in mice in treating a form of deafness were reported just earlier this week. So what's your general sense about, do you anticipate that in the next, say, 5 years, base editing is going to be right up there with more traditional CRISPR genome editing in terms of clinical applications?
Well, I think clinical applications is certainly, as you said, one of these things that we're looking forward to in the future. In addition to the paper you mentioned, there were 4 other technical improvements papers, I think, simultaneously published in the last week. And I think that's where things are very exciting. Early on in the days of zinc fingers, there was kind of a small number of papers that would come out in the time period, whereas it's just been an explosion in the last 5 years for CRISPR-Cas. Similarly, and a bit delayed though, we're seeing a lot of groups jumping onto base editing and other related CRISPR fields. And so I think it's exciting to keep your inbox open to this flood of papers that we see announced. And I think as we move forward in base editing, we'll address some of the hurdles that have been identified, and it'll be exciting to see how these can be used in certain disease indications, and as I had in the one slide, possibly in conjunction with CRISPR-Cas nuclease editing.
Yeah, great. Thanks so much. Just absolutely flooded with questions here. So we will work with TriLink to try to get those questions cataloged and in your hands, TJ. But we'll do a kind of a CRISPR Gen webinar overtime here. We'll just go for another 5 or 10 minutes and see if we can get through a few more. Can we target mitochondrial DNA with CRISPR?
Yeah, I think that's a good question, and one that we often talk about, looking back at what was done in previous systems and thinking about how one gets zinc fingers or CRISPR-Cas into the mitochondria. And there's a range of different technical challenges in terms of repair and getting it in, but it's certainly one thing that has continued as an area of interest, and so I think that's something one could look for further publications and research in that area.
Okay. Off-target effects may not be a big deal for cells using drug screening, but what should the release criteria be for gene-edited cell replacement therapy?
Well, I think that there could give a whole talk on how one does off-target analysis, and I certainly have at different times, and certainly this is one of those areas that on which I continue to consult and advise. And so since we're limited in time, I think I'll hark back to the discussion of risk-benefit analysis. And so as one weighs different diseases, weighs alternate means of treatment, one could look forward to diving in and trying to understand as best you can. And I think that what doesn't get relayed sometimes is that we've done a tremendous amount of work before we even get into the cells to understand the possible off-target sites. And I talk about the bioinformatics. Our website, COSMID, is still up to allow people to do that. And once you start going into cells, you can order the primers that COSMID designs or design your own and begin to take a look at the probably most worrisome or putative off-target sites to see if they're significant. By the time we would move towards the clinic, we've done a tremendous amount of sequencing and assays. And so when we're unable to detect anything for some of these lead target sites, it's very encouraging that we've kind of raised the bar in how deeply we've looked for off-target effects. And that certainly is part of that risk-benefit analysis. And it just is limited by the level of detection.
Thanks. Darren, who's in startup land, says, where do RNPs come into the equation?
And certainly, since CRISPR-Cas has been kind of readily used initially in plasmids, as I described from 2013 on, it was exciting to see that a number of vendors are making the proteins. So in days of old, you'd have to call up your good buddies at neighboring labs who worked wonders in proteins and ask them to try and make something the size of CRISPR-Cas, as Cas protein is fairly large. But now, as readily as one can order EcoR1, one can get the protein in and thereby add the guide to the protein and put that directly into cells. And one of the reasons that that's important is that the protein isn't around as long as plasmids, so that also helps improve the activity and limit the amount of possible off-target cleavage effects that might interfere with your research.
Great. Renata, who's a medical student from the University of Groningen. Hi, Renata. Thanks for the question. What's the most commonly used delivery method for CRISPR-Cas9 into cells, and which one is the most applicable for cell therapy?
So certainly we use a number of different means going into the cells, and one of the effective ways for treating cells ex vivo is even electroporation. And so there's a good bit of knowledge on how to get things into the cells, how you keep altering the protocol because one wants to keep the cells, if the term you want to use is stemminess. And so it provides an effective way to get the Cas reagents into the cells. One might want to put in a donor with AAV, and certainly people are continuing to work on both viral and liquid nanoparticle means of getting into the cells for ex vivo, but also in vivo use. Medea, if I may not be pronouncing that correctly, wants to know, do you teach working with software to designing guide RNA and selecting PAM sites, etc.? Maybe, maybe you have a recommendation. What are some of the most reliable software tools that you might even potentially, if not endorse, at least suggest people check out? Oh, there's certainly a wide range of different programs available. I did do one before it was so easy to do. Made a quick video on how to use bulk processing with our COSMID program. And the question is, if you, at least that program designed primers for you, but if you're making a whole lot of targets, you might want to invest in trying to, as this question is suggesting, better learn some of the many different programs. I'm beginning to work with a few companies that are hoping to address this question and try and make this more readily available. But there are a number of our programs already available that could help in this. If the other part of the question was, is there a good resource, a webinar precisely on that? That is a very good question, and I unfortunately don't know of a good resource in that means. Okay. James wants to ask,
could you comment on the use of vectors such as AAV to get expression of telomerase? Ooh, interesting. I understand that this may add plasmids rather than change existing DNA.
Okay, so sorry, I'm trying to look through all the pile of questions here to get that. The question was, can we use AAV as a means of delivering a particular protein, in this case telomerase, and there was
I think so.
a hope to replace now I finally found it, to maintain the survivability of the transplanted cells. Oh, I see. Yeah, I mean, I think that there certainly would be interest in adding many different reagents and components and small molecules that affect the DNA repair pathways. And certainly people are looking at that to try and drive intended repairs. I'm not sure if there's been any effort to try and look at putting in telomerase in order to increase the livelihood of transplanted cells. So I can't really speak too much to any of the methodology with telomerase.
Okay, thanks. Ramaro wants to know from NIH, are there any studies to decide the appropriate time to add homologous sequences with CAR after CRISPR-based gene cutting?
There's another one where I'm going to try and determine which of the two kind of relevant questions there. There have been studies that determine how much of this homology arm one needs, and obviously one can optimize that based on how big your cargo is. But if one's trying to put the CAR complex in, that is an important facet because there's some kind of trade secrets and maybe unpublished work that's looked at optimizing, let's say, the inclusion of a viral donor along with your cutting reagents. And the timing goes back to some of the early work which looked at putting these things in with plasmids versus protein or mRNA. And so I think that as the take-home message I put forward is I would look to optimize that depending on how exactly you're getting the reagents in, when you would have the active proteins, and want to have the donor available for homology-directed repair.
TJ, good question coming in at the end here, which I will just read it from Razor. Do you imagine any application for CRISPR in the treatment of COVID-19?
Oh, there certainly is a lot of interesting work being presented on a range of different aspects of that. There are people that are making models by either inserting or making changes to ACE2 so that you can say, can we model this in mice better? There are screens that are identifying drug targets using CRISPR-Cas. There are several different approaches that are looking to have put together diagnostics for COVID using CRISPR-like systems, and I think those are very exciting. There have been some discussions of targeting RNA, and so I think that's an interesting aspect, but I think it's less straightforward than other instances where one wants to correct DNA or target a virus that has a DNA intermediate. So I think that there's very encouraging diagnostics and modeling, and that might be what we see in the near term is the very exciting contribution by the CRISPR-Cas community.
Yeah, there was a paper published in the last few weeks by Stanley Chi's group in Cell on a technique called Pac-Man, using Cas13, if I'm not mistaken, to try to directly target the virus. Obviously, a very long way to go before you could even conceive of using that in actual patients, but some promising, if preliminary, results on the therapeutic front. I think a related question from Gaurav, why can't we use Cas13 instead of Cas9 to treat cancer?
Well, I guess one could make CAR T cells using a wide range of editing techniques. Certainly, once you've come up with a set of nucleases, whether it's Zinc-Fingers, TALENs, CRISPR, for making your initial edits, let's say knocking out TRAC, inserting the CAR complex, etc., then one can continue to use those. Certainly people are using formerly known as CPF1 or other orthologs of Cas for editing T cells. And so if you're talking about how one makes chimeric antigen receptor expressing T cells of various types, it's certainly within the realm to use any of a range of different nucleases in addition to the ones I just described.
Great. Well, TJ, I think we've got, as I said, a wealth of questions, but we're going to have to wrap things up now. I apologize if we couldn't get to your question, but we've got them all captured here. And with our sponsor TriLink, we'll make sure that TJ, we'll try to figure out a way that he can respond. TJ, just in the last 30 seconds, so are you going to hang out being a consultant? You seem to be enjoying the life as a consultant because it gives you sort of free range to talk to a variety of different companies and do some cool stuff. So is that where you're going to hang your hat for a little while?
Well, I was just about to comment. Wow, all these questions that'll give me something to do for the next bit. But I think it's very exciting because there's a number of best practices and means to continue to move forward. And so I've enjoyed the discussions. I've enjoyed the companies that I'm working with, and because it's certainly there's a lot left to do because it's so exciting to look at the potentials, and many of the questions, trying to click through them all here as they roll in, address some of the different disease indications or different technical aspects that we could improve upon. So it's an exciting time to be in the field in general, and also to be in a place where you can help a large number of groups. Yeah.
Well, if you've got time on your hands, you could think about writing a review of all this stuff for the CRISPR Journal, but we'll talk about that offline. Thank you so much. Thank you, TJ Cradick, formerly of CRISPR Therapeutics, and as you've all heard, just a fantastic font of knowledge and experience in not just CRISPR, but all forms of genome editing. And I'm sure it'll behoove us to try to figure out another way to get him in front of you to share some of his insights with you. So thank you, TJ. For folks who wanted a certificate of participation, please just email us offline and we will get you something that signifies that you were present and participating throughout the webinar. And as I mentioned earlier, this is part 2 of a 3-part series from TriLink. You'll find this and a whole host of other webinars on demand at both the CRISPR Journal landing homepage and the Genetic Engineering News webinar page that you'll find on genenginews.com. So you can just a trove of CRISPR-related genome editing and other webinars that you can view on demand for free. But thanks to TriLink, and part 3 of this series will be coming up in the next few weeks, so stay tuned for announcements on that. Most of all, I would like to thank you for sticking around. Apologies for the little technical pause in the middle there of the Q&A, but we're glad we could reconnect with TJ and get through so many questions. So we hope to see you all again very soon for another CRISPR-related webinar, either from the CRISPR Journal and or Genetic Engineering News, our sister organization. It's been a pleasure to bring this to you. Thanks to my colleagues at GEN and WorkCast, in particular Jeff Rogaliskis and Nina Park. And for the whole team, I'm Kevin Davis. Goodbye for now.